In the not-too-distant future, a revolution is brewing, one that will change the very fabric of our existence. It's a tale of cells that hold the key to unlocking the mysteries of life itself. Let us venture into the unknown, for in this age of stem cells, anything is possible.
I've seen some beautiful flowers.
I noticed the mug and the hat and everything. I love it.
I just got back from spring break.
You look adorable.
I have returned inspired. You know, the last time I went to Disney World was when I was 8 years old. And I remember leaving feeling like anything is possible. You know, because to me, I saw like impossible things. And so our twins are 6 now. So I thought this is a perfect time to go before there's like nothing surprises them and they're totally jaded, you know. So we took them, and I'm sure it was a magical experience for them. But for me, again, as an adult, you know, the parks really haven't changed. Like the rides and everything, same things that we did when I was little are still there. Like It's a Small World, Peter Pan, all these things. But they've implemented all this technology, and it elevates everything. It just makes you feel like a kid again. And so there's no place more magical. And it's a testament to the fact that in America, we have the most creative minds. We absolutely do. It's a really great representation. So have you been?
I have not.
You have to go one day, Jan. You have to you just have to save those dollars, and you got to go. And anyway, I come back with this feeling that we are in an exciting era. A lot of changes are happening in the stem cell world too. But I thought that this is an appropriate theme for the magical world of stem cells that we're going to talk about. We are on our our 4-part series on the regulations of stem cells. And today we're going to be talking about the magical effects of systemic use.
Well, we've talked about minimal manipulation. We've talked about homologous use. We have affirmed thou shalt not mix, right? And so this is the last part of that set of regulatory definitions that really impact, our access, everyone's access to cellular therapeutics. So we're grateful to everybody who's joined us on the journey thus far. And if you hung in, now you get mouse ears in the final episode. So it's totally worth it.
Systemic use is if you just take it for its words, you would think, like in the system, throughout the system, like blood is flowing throughout your system. So something that's not localized, but in your entire system. Now, I can give you some history as to how this even became a thing, because when you read this, instinctually, your mind first goes to, IV stem cells, because intravenous stem cells is a real big hot topic now. Intravenous stem cells probably started close to 20 years ago. I do think I was one of the first people to receive IV stem cells. I did so in a lab in South Korea. And we weren't supposed to do this, but we were a bunch of lab rats and we were doing this. And it was because we were able to culture cells. So being able to culture and grow cells, people say, how could I have experimented on myself? Well, I saw the cells, so I knew what I was putting back into my body, and they were my own cells. So I felt comfortable doing that. But receiving cells in large doses, nobody had ever done this before because we didn't have robust culture technologies. We were always dependent on whatever we could isolate from the body, which were small amounts of cells. So if you have small amounts of cells and you have a brain condition, for instance, They would drill holes and put stem cells into the brain. The ability to culture cells or expand the number of cells opened up a whole nother world of administration by IV. And the idea was, if the stem cells are naturally going to navigate to areas of injury in your body, then giving it systemically is a great way to administer these cells. The only problem is, if you don't have a lot of them, then the amount of cells that get stuck in your lungs, because the lung is a giant filter, then it wouldn't be able to have a chance to circulate, because after the lungs, it goes into the heart, and then the heart pumps in and circulates it all over the body. So if you don't have giant amounts of cells, and they also can't be too big, and during culture they get big and they get sticky, anyone to cell culture knows this, then you get like clumps of cells. Well, then those really get stuck in the lungs, and those don't go anywhere. And so giving cells systemically, you have to give high quality, high amounts of cells. And this wasn't a possibility up until, I'd say, like 15, 20 years ago. So I think, I mean, it wasn't documented, but I think I was one of the first, people to do that. And it was really just to see what would happen. Like, is this safe? And I along with a few other people, we all got these IVs, and nothing happened. Nothing felt different. But I will say that I had plaque psoriasis for a very long time. And I had it mostly in my torso area, but pretty bad plaques on steroids, on prednisone. And I remember after getting cells, and it wasn't instantaneous, because it didn't occur to me until about a month after I had received my first and only dose that I wasn't itchy. I think I was like 24 at the time, and I wasn't itchy. And then a few months went by, and I still wasn't itching, and my plaques were starting to disappear. And then soon enough, I was just totally normal, and I've never relapsed since. So at the time, I never put those two things together. I just thought I grew out of it. It isn't until now, where we understand all of the biology behind these cells and how they work, to know that I was able to put two and two together. So that's my story of IV stem cells. But the law as written does not just encompass how their cells are administered systemically versus locally. The actual definition, FDA's definition, is what?
FDA defines a systemic effect as a biological action that extends beyond the site of administration or affects tissues and organs beyond the site of administration. So it's something that has a systemic effect.
So how do you control cells? You put them into the body? How do you box them in?
Well, it certainly seems at odds with the nature of what we know to be true about a cell, right? That a cell has an inherent capacity, a living cell, to affect and impact an entire system, which is part of the inherent promise, the hope in cellular therapeutics in general, is that we have reason to believe they can go into the body and do things that other modalities can't, that they can do things that perhaps we don't know about, because we know they have an ability to target and hone in on a site of injury or illness or disease, oftentimes that we don't even know existed. I mean, I think continually of in one instance where an individual received the cellular therapeutic manufactured here, right? He'd been living with Parkinson's. He was in his late 80s, been living with Parkinson's for a very long time. And he experienced a clean EKG for the first time in 40 years after several cell therapy treatments, right? It stands to reason the only thing that changed in his living routine and anything, the only thing that changed was receiving stem cells, right? So clearly the cells seem to think that that was an area of concern. I use the term think loosely, but the point is, yes, it seems at odds with what we know to be the nature of cells as they function as cells in our bodies.
So speaking of function, this regulation also says that it cannot have metabolic activity. So it cannot rely on the systemic effect or the metabolic activity of a living cell. Well, if the cell is living, my question, I guess, to regulators, if any regulators are listening, is how do you have a living cell without metabolic activity, period?
Well, and one consideration of metabolic activity under this set of regulations is the way that the therapeutic interacts with the recipient's tissue. To which I ask, well, isn't that the point? Don't we want it to interact in a productive, healing way? There seems to be a pretty foundational disconnect here.
And the metabolic activity of a cell is often dependent on the host condition. So where the cell is placed. For instance, how the cell behaves in a lab on a petri dish is different than how it behaves in an animal, and different in the way it responds in a human. That's why a lot of the regulations on doing animal testing is going out the window, because they're saying we should test more on humans because humans are not animals. Yes, right. And we're not petri dishes either. And so the way cells behave are inherently dependent upon the environment that they're placed in. But again, how do you stipulate that they don't have metabolic activity? This, I would say, trumps everything. Like, we don't have to be concerned about minimal manipulation or homologous use or any of these stuff. They're saying the cells can't have their inherent metabolic activity. In my opinion, which doesn't count for much, but in my opinion, metabolic activity directly correlates to potency of a cell. Potency, stem cell potency, is this big hot topic in the world of developing stem cell therapeutics, because in order for FDA to approve your product, you have to actually show that your product is potent. So how do you do this? Most of the time, what has been accepted, general acceptable measures for potency are measuring what the cell secretes. Okay, so if the cell secretes X amount of protein that we think is helpful in this disease, then we can check that box off that we have created potent cells. But where are you measuring that? You're not measuring that in the body, you're measuring that in the petri dish. So we're going to go through all this time and effort to measure what is secreted in a petri dish, but we still don't have the answer as to whether that is actually happening in your own body, and nobody can answer that. You know, yesterday I had a really great conversation with a very prominent doctor, researcher in the Med Center. He was here, and the conversation got into cells and functions, and he knows all of this very well, the biology very well. And I think we actually have different religious perspectives too. But at the end of the day, he mentioned, well, you know, I just don't have an explanation. It has to be intelligent design. And that is, I think, the best explanation, because we cannot explain why a cell does these things in the body. Case in point, rheumatoid arthritis. We did a clinical trial for 15 patients with RA. They all had very severe RA for many, many years. They had about, on average, 15 to 20 joints that were affected. And FDA at the time only allowed us to give them one infusion, one IV infusion each. And after monitoring them for an entire year, almost all of them had no signs of RA. So even though there was no placebo in this group, it was statistically impossible to have achieved that kind of a result, unless we totally made it up.
You know, there's a fair and growing amount of research on MSCs and RA out there in the world, and they've not seen the same results that you're describing with one exposure yielding this kind of prolonged effect. So how do you account for the difference?
Well, good point. I mean, it took forever to get through peer review, and it was precisely that. It was trying to explain the discrepancy behind, and this is why not all stem cells are created equal. I'm not saying that ours are superior. They are. But it really comes down to, first, I submitted a picture of all 15 patients' stem cells. Why did we do this? I submitted a picture of all of them to be able to show visually all 15 patients got the same product. So until you can, if you want to achieve similar results, first you have to give a similar product. But now is the most important characteristic of the cell is, are they alive? Are they viable? Are they metabolically active? That is another key measurement. And so we provided that table. And we knew that this was actually going into the person because we didn't freeze these. Frozen cells weren't going into these people. We measured this right before, the day before they were actually going to receive treatment. And we had already validated and knew that the stability of the cells was good. So we knew that what was going in was close to what we had measured. And so when we saw the result from the trial, we knew that if you create consistent lots, despite the fact that we all have different cells, and arguably, if we all have different cells, then we all have different metabolically active cells, right? They're not all the same. But again, going into all of those different bodies, and despite all the conditions that they had and secondary conditions they might have had, I mean, who knows? They might have had underlying cancer or heart issues or whatever it is, along with their RA, the cells were still able to navigate through those body conditions and still achieve a consistent result. That was what was so novel about that study. But anyway, even though you achieved that result, I still can't tell you how the cell did that. I know very well what MSCs do, like their behavior, but what pathway did they take? What specific proteins did they secrete to achieve the clinical effect that we saw in RA?
No one knows.
Nobody knows. Only God knows. Yeah.
Only God knows. Well, and I think that's part of the philosophical commitment that should be made for a cohesive set of regulations to result, right? It also strikes me when we talk about metabolic effect, in the loosest sense, degenerative disease can be thought of, one facet of degenerative disease can be thought of as, there's a reduced volume of stem cells just over time as we age, right? And part of halting or reversing that is introducing more cells into the body. So another way to think of that is there's a decline in metabolic function of the cells. We can rectify that to varying extents in certain circumstances by introducing increased metabolic activity. If we think about it that way, then this regulation is essentially saying anything that might actually help, we're going to regulate as a drug.
Well, you see the fault in that, because if FDA's role in establishing safety and efficacy of a product is to do that, then there has to be parameters that we can all control. Like, for instance, minimal manipulation and thou shalt not mix. Those are functions in the lab. Those are controllable functions. Okay, don't manipulate. Okay, well, tell me what that means, and I won't do it. Right? Those are in our control. Those are things that we can actually follow.
Yes.
But like non-homologous use, homologous, and this systemic effect or metabolic activity, again, you cannot have a living cell, a living healthy cell that does not have metabolic activity. You cannot have those two coexist. It's basic biology. So
Yes.
how can you control that? Then that means that no cell is allowed. But they are.
Yes, there are exceptions to this set of regulations, and let's talk about them. I mean, one that leaps immediately to mind that most folks are probably familiar with. What about blood transfusions? You know, blood is comprised of living cells that go throughout the body. That's what we want them to do. They have a systemic effect. They affect your entire body. So why aren't they regulated like drugs?
So blood is our human cells. If you take blood from another human, that is a human cell. But they don't fall under HCTP. They're like a separate category in and of itself. And it's actually because HCTP regulations didn't exist when blood transfusions were happening. Yes.
Well, and that explains too how, you know, blood transfusions get around another subset, right, which is the autologous use consideration that cells are to be taken and used in the same individual. Well, that would preclude blood banks. I mean, anything that we commonly see in most of our communities, you know, this blood transfusion functioning in our daily lives as in emergency medicine in particular. And guess what? It's been such an asset to people in need and people who are sick.
Oh, of course. I mean, it is a life-saving mechanism, even in the trauma world, which, side note, we are going to CTAC in early May in Arizona. And so we would love to see you all there if anyone is planning on attending. We are going to take the podcast to Arizona. Did Taylor know that? Did you know that? Okay. So we are, anyway, that's total side note. And that's all about trauma, blood products, stem cells. If you're interested in that world, it's an excellent conference, and you'll get to see like real life use on the battlefield and in clinic, and get a good perspective on what is going on right now. But that was total tangent.
Well, but it's not such a tangent, because I think one of the takeaways that's pertinent to this conversation is that this is an example, a real world example of a technology in which a biologic functions in a different class of regulation, neither completely a drug nor completely an HCTP, right? Here's an effective way to categorize it that's something unique to it and honors the biological nature of it. Mm-hmm. Why can't we do that with cellular therapeutics? We also do it with reproductive technologies.
Right. And so it is like the time that it was invented. Timing is everything. You know, they tell you timing is everything. If blood transfusions didn't exist until now, and we decided to, do them now, under this set of regulations, we wouldn't be able to. We would be stuck because it won't fit. Again, why were these regulations created? I think it goes back to tissues and organs. That's really what it was for. The safe transfer of tissues and organs and products like that going from one person to another. Just make sure that it's donor tested, make sure that it's free of viruses, make sure that it's handled properly in the lab and transferred under the appropriate temperature conditions, things like that. I think that's what this was for, because it clearly you can't stuff any kind of stem cell therapeutic into this mix. That's the bottom line. If metabolic activity is one of the criteria, then there's no way it can fit. Now it says so they're allowing systemic use if it's autologous. But again, why are autologous stem cells, my own stem cells, not allowed to be infused back into me by IV infusion, unless it's a drug in the United States, is because manipulation, more than minimal manipulation, homologous use, for those reasons. So you get caught in the other regulations, even if it's autologous. Yeah. This
Sub-FASA regulation, though, to me is the one that most intuitively, if you've never considered a scientific definition, if you've had limited exposure to scientific rationale, still leaves you going, huh? Because if you accept that the body functions as a whole, that the systems within the body interconnect and affect and impact each other, then by default, introducing something like a cell with an inherently metabolic function. I mean, you memorize that in middle school biology, right? That cells have a metabolic function.
Even their ability to divide is a metabolic.
It's a metabolic function.
Right?
Yes, that's what they do.
That's what they
They're
do.
cells. They're cells.
And they're going to poop stuff. They're going to eat stuff and they're going to poop stuff. That's the secretions. Those are the things that they naturally secrete. And so they're saying metabolically active cells are also going to secrete stuff. So they can't secrete stuff either. That means that the only things that qualify would be dead cells. Anyway, that's another conversation.
And that is another conversation.
And there are people who believe in dead cells. We're not them.
That doesn't seem to pass the common sense test to me either. And I'm biting my tongue so that we don't go down that rabbit hole. But again, it's, a lot, if you look at this from a common sense perspective, I think is often the most fruitful, because the people sitting in the chair, which maybe one day it's us, who need access, who deeply desire and seek access to promising, proven, safe regenerative technologies, including cellular therapeutics, right? If you can look at this and their reaction is, well, wait, that doesn't make sense. I actually need something that will affect my entire body, and I need access to it. How does slowing and hindering access through regulation as a drug help a person in need? I'm not convinced that it does. Whatever the intentions of this set of regulations might have been, from my perspective and my point of view, it's beyond time to revisit them in a way that honors the nature of what we're studying here.
Well put. I think, helping people in need, that of course is the main goal behind at least why we operate. And I hope other people who are in the drug development world are also with that same mindset. There are populations of people that we need to help right away. Mm-hmm. The process in which we're going about this, whether it be the drug route, so Hope Biosciences is going down this drug route because we don't meet these criteria. So clearly, again, our cells are alive. The cells we give back are truly alive and well, and definitely metabolically active. And they're going to be introduced systemically. So we don't meet any of these HCTP categories, and therefore we are bucketed into the drug world. But a drug has to have a clear pathway. At least that's how it has been in the drug world. Once you prove that your compound, your chemical, whatever it is, provides a certain function in that disease process, you have to prove that what you make is that compound. But again, in the stem cell world, it's so hard to put it in a classic drug category because it doesn't behave like a drug. It is a drug with many hats and can evolve depending on the situation. So how can you put it in a box and say you have to behave like this and you can only do this? You can't. You just can't. It's biologically impossible to do that. There are actually people who are trying to put a more strict definition on what the cells do. So for instance, you take a stem cell that can do all these things, and if the disease you're treating is, let's say, liver cirrhosis, okay, and you're trying to make new liver cells, then rather than use the stem cells as they are to regenerate the liver and do all the different functions it does, you actually in the lab differentiate those into liver cells. So it's a finalized cell type because the function is your liver doesn't have good liver cells. So we're going to put good liver cells into that liver. It's just very simple. That's the mechanism. But time and time again, we've seen that, again, it goes back to systemic effect. The way your body heals is not ever localized. It may appear to be localized, but it isn't. People with like diabetic foot ulcers, right? It's a circulatory problem, right? Diabetes causes circulation, poor circulation, and you lose circulation into your lower extremities, and then you don't have blood flow. And so when you bump into something, it becomes untreatable and just gets worse and worse and worse until you have gangrene, and then you have to cut off, you have to amputate. That's the only solution. For a long time, you took these black, rotten extremities, and when we would look at that, we would say, okay, we should put stem cells in that area. Why? That's the logical way to treat something, right? Okay, that foot is black. Like, let's try and save that foot. So you put stem cells around that area. You sprinkle them on there, or you inject them subcutaneously. You do whatever you have to do. Well, now these things are treated by IV. Why? Because the stem cells go in intravenously, and they are known to produce new blood vessels. They actually make microcapillary. When there is a traffic jam in your circulatory system, they are going to make little capillary, because they know blood has to get through. And they dig their way through and create those new avenues for blood flow. And then soon enough, that foot is good as new. I have seen black, completely rotten tissues come back to life. And that is the power of the body's regenerative system. And it's a system. So it is systemic, because healing starts in the place that's least expected.
Well, and it strikes me that, in so many parts of these regulations is reflected the presupposition that we know even how the body works. Mm-hmm. And we don't. It's, and we started with this discussion of magic and feeling wonder. And there are two ways to approach the assertion that we don't know. One is to clamp down on it and say, yes, we do, and develop a set of regulations that reflect what I believe to be a false assumption that we know. The other is to go, we don't know, and to allow room in our regulatory framework that, while respecting the safety of the people receiving investigational therapies, acknowledges we don't know.
It's the magic of our bodies.
Yes.
And the magic of our cells. And we can't take credit for that at all. That is what has been placed in our bodies to do what they naturally do. And just as we take an organ from one person to another, and that's logical to us, cells, the building blocks of all of those organs, should be considered to be the same. They are regenerative tools. They're not drugs with one-dimensional pathway to a specific symptom or problem. This is a totally different, again, unexplainable cell gift in our body that we are trying to develop as tools so that everyone can use them. And doctors are going to be really critical in this piece, because doctors understand, and all the specialties and researchers, brilliant people I have seen along the way, understand their field so well and understand the limitations in their field. And once their eyes are open to what these cells can do, put those things together. And it's beautiful to see, because we are doing some first in human cases of all of these different problems. I just got an email from the mom of our patient at the last compassionate use patient that we had for epilepsy. He was having 30 grand mal seizures a month, down to one. And both actually that happened may have a causal effect, with a different treatment. And so, and they call these kids FIRES kids. They're drug resistant. They've tried every drug out there, and tried everything, and seizures still return. And I remember talking to a researcher at Texas Children's. He's since retired, but he was telling me that there is no solution for this group. There's absolutely nothing you can do for this group. And that if there was something seen in a child like the one that we treated, that it would be a true miracle. And I think we've witnessed that. But now to translate that into a trial will do, and hopefully will show the same effect. But at the end of the day, nobody, doesn't matter how smart you are, how long you've studied biology, we will probably never get to an answer as to exactly how these cells are doing what they're doing. And so we just have to accept.
Well, and we'll certainly never progress toward an answer if we clamp down on it, right? And I think that every facet of this conversation about regulatory definition has brought us to the same conclusion. There's a real need for a different regulatory class, right? Something that puts parameters in place that does protect people, especially, as we know, there's a lot of variation in our field, and it does need to be above board. But it's not as a drug.
Okay, so then maybe for fun, our next episode, we talk about the solution. What's our proposed solution? And we had a good one. We came out with a long time ago. We had a pretty good one. We drafted a long time ago, and still a relevant conversation today. So that'll be very, very interesting. In recent events, Dr. Peter Marks, who's the head of CBER, he is now gone from FDA. And so far, we haven't seen a lot of changes in our communications with the agency and everything, and all the things that we're doing. But changes are coming. In a recent interview, he did say that stem cell regulation changes are happening, which I think he's not in agreement with. I'm curious to know what those are. Mm-hmm. So amidst all of this discussion, we might get some news. Yeah.
Yeah, there's a lot to talk about. One thing I'm encouraged by is that what we thought was a solution 5 years ago when we initially sat down to draft that proposal, I still think is a solution, right? And so that in and of itself is a hallmark of validity that, let's talk about it. Can't wait.
Okay. All right. Well, until next time. Bye.
Bye.